Hello. In this series of lecture, we are going to look at protein synthesis, and we are going to start by looking at transcription. As we have learned previous in the class, we learned about the central dogma of molecular biology, and we saw DNA is replicated to form another copy of DNA, and DNA can be transcribed to give us a messenger RNA, which can be translated to give us proteins. So in this particular class, we're going to look at the process of transcription, and we are going to focus on bacterial or prokaryotic transcription, especially of E. coli, because it has been well characterized. At the end, we're going to summarize on the differences between prokaryotic and eukaryotic transcription, and we are going to focus on important details present on eukaryotic transcription. So to remember these lectures, look at this to remind ourselves of the differences between DNA and RNA. So the first difference we had mentioned was that RNA, the difference happens in the ribose sugar, where the RNA has a hydroxyl group at the 2' position, and that's why it's called ribose. And while the DNA, the OH group the hydroxyl group has been replaced, and we have a hydrogen group and refer to it as deoxyribose. That gives it the name deoxyribonucleic acid. Another difference is in the bases. In RNA, we have an uracil instead of the thymine base. I think this one you remember. Other difference is that while DNA exists as a double strand, as we can see in this picture, a duplex, most RNA carry out their functions as single-stranded. The single strand might fold itself, as we are going to see in transfer RNA, but they mainly exist as single strands. We're saying here RNA strands can fold back on themselves, and they have the potential for much greater structural diversity than DNA. Remember when you describe the structure of DNA, the way it's a duplex and then it is folded, especially in eukaryotes around histones, reduces the ability of it to have structural diversity. RNA molecules are mainly involved in storage and transmission of information, as we've learned this in class. And recently, we found out that there are RNAs that have catalytic activity, and they are referred to as ribozymes. So what is transcription? So transcription is a process of which information from a segment of DNA is copied into an RNA. And as we know, there are three major types of RNAs. We have the messenger RNA, which encodes amino acid sequences of polypeptides, which are specified for a particular gene, or gene set. So the messenger RNA carry the information on DNA of a gene, and they carry it to the ribosome for translation into proteins. Then we have transfer RNAs, and the transfer RNAs are able to decode or translate information coded in the form of codons into amino acids. So that's why we are saying they're able to read information encoded in the messenger RNA and transfer the appropriate amino acid to the growing polypeptide chain during protein synthesis. We're going to see this in the next lecture of translation. Then we have ribosomal RNAs, which together with proteins form our ribosomes. You can see here. But in addition to these three major types of RNAs, we have other types of RNAs, such as small nuclear RNAs, and we're going to look about them as we encounter them. We're going to talk about them as we encounter them. So there are similarities and differences between replication and transcription. So remember, replication is where we copied DNA to form another copy of DNA, and then transcription is from where you get the information from DNA to form an RNA. So for both of them, replication and transcription, will require a DNA template. And also both of them involve polymerization reactions, where nucleotides are polymerized in phosphodiester bonds. And in both of them, the synthesis of the new strand, whether it is a DNA strand or an RNA strand, is synthesized in the 5' to 3' direction. And similar to what we saw in replication, we note transcription also has initiation, elongation, and termination steps. However, Transcription, unlike replication, does not require a primer. We saw replication required an RNA primer, which was later on removed. But transcription, as we are going to see, does not require a primer. Interestingly, while replication involved copying the whole chromosome, in transcription, only a gene or a gene set is involved in transcription. Okay, so not the whole chromosome is copied in transcription. And also only one strand is used as the template. This is what we call the template strand. The one that is copied is called the template strand. And the opposite strand that is not copied, we refer to it as the coding strand. Okay, so this is a major difference because in replication we saw both strands were copied. So the enzyme that is involved in RNA synthesis is referred to RNA polymerase enzyme. And as I mentioned earlier, we're going to mainly focus on the transcription process in eukaryotes, especially the E. coli. So the RNA polymerase enzyme is able to synthesize RNA complementary to the template strand of the DNA. So it requires, of course, the ribonucleotides, the ATP, UTP, CTP, and GTP. Note here is UTP and not TTP. And also note they're not deoxy as the ones we saw for the DNA polymerase. In addition to these ribonucleotides, it also requires the cofactors, magnesium and zinc. So, similar to DNA polymerase, RNA polymerase also adds nucleotides to the 3-hydroxyl end of the RNA strands, which will be growing in the 5' to 3' direction. And it uses the Watson and Crick base pairing interaction. And the only difference is that instead of T to A, T binding to A, we have uracil binding to adenine, and still the same guanine binds to cytosine. So during initiation of transcription, the RNA polymerase is able to bind to promoter sequences on DNA. We are going to talk about these promoter sequences. And unlike DNA polymerase, the RNA polymerase lacks proofreading activity. Hence, there's a higher error rate in the transcription of proteins. However, we know these RNAs, many of them are destroyed immediately after translation. And so these errors are not likely to be transmitted for generations, as we see that might happen in replication. So although the error rate is a bit higher, the consequences are not as dire as if these errors happen during replication. So similar to what we saw in replication, we saw a replication bubble, but also in transcription, we have a transcription bubble. So this bubble is formed from the unwinding of around 17 base pairs of the DNA duplex. This unwinding is necessary to allow the RNA polymerase to be able to access the DNA template and to be able to synthesize the new RNA in base pairing with the template strand. So in this replication bubble, we have the RNA polymerase unwinding a section of the DNA of the double helix. And we have a short DNA RNA hybrid of around 8 base pairs, which peels off because the interaction between RNA and DNA is not very strong, especially A and U, adenine and uracil is not very strong, and within a short while it detaches. So even though the section that is open is around 17 base pairs long, only 8 base pairs are involved in interaction between DNA and RNA. So we know, of course, as the DNA helix unwinds, we end up having supercoils, either in front or behind the section of the transcription bubbles. And these supercoils are relieved by the enzyme topoisomerase. We saw it in replication. So the topoisomerase is able to go and relieve the supercoiling that happens because of the uncoiling of the DNA to allow transcription. We mentioned promoter sequences. We say that at the beginning of transcription, the RNA polymerase binds to promoter sequences. What are these promoter sequences? These are sequences in DNA which are found upstream a gene or in front of a gene. Okay, and these serve as pointers or initial binding points of RNA polymerase And in terms of in eukaryotes, they also point as pointers for the binding of transcription factors. Okay, and they can be located -70. When we talk about negative, we mean it's 70 base pairs before the gene of interest, up to 30 base pairs after the start point of the gene of interest. In prokaryotes, they include the Pribno box. So the Pribno box is very close to the gene of interest. For example, if we look at this, this is where the gene of interest starts, where it is written RNA start. Now, at around 10 base pairs before the start of the gene of interest, we have a sequence which we refer to as the Pribno box that has 5' from 5' direction TAT to 3'. So this is what we refer to as the Pribno box, and this site is where the RNA polymerase will start unwinding the DNA. You can see the DNA is unwound quite a bit earlier, before the point of the start of transcription. Another promoter sequence that is recognized by RNA polymerase is at the -35 region. So this is the initial point of contact of RNA polymerase with the DNA, okay? And its sequence is 5' TTGACA. So these sequences are recognized by the RNA polymerase, and it is able to bind here. There was also another region which is referred to as the UP, or the upstream promoter region, which is located around -40 to 60 base pairs upstream the gene of interest. And this one is present only in highly expressed genes. Okay, highly expressed genes, or the genes that are used regularly in the cells. For example, in a cell that is breaking down carbohydrates, you'll find the genes for glycolysis here. Okay, so these sequences are referred to as consensus sequence because they are highly conserved during evolution, and then they are recognized by all RNA polymerases. So we can see, like in this diagram is showing here, all this is what we refer to as the promoter DNA. It contains sequences that allow RNA polymerase to bind before the start point of transcription of the gene of interest. So what happens? We said a transcription also has the 3 steps: initiation, elongation, and termination. What happens in the initiation steps? So in the initiations, we can think about it as consisting of 3 steps. So the first of it is binding of the RNA polymerase to the promoter, okay? And we have said there are 2 major sites at -10, the Pribno box, and -35. So the RNA polymerase will bind. Initially, it will bind to the closed DNA. It's able to recognize these sequences in the double helix, and it will bind in that double helix when it is closed. But it eventually opens, separates or melts the DNA strands at the -10 base pairs, which are the Pribno box, and then transcription starts at the separated DNA strands. Okay, so we can see this initial binding to the closed complex, then we have opening the complex to create our transcription bubble, and then DNA transcription starts, and then the transcription now moves along the unwound region. The next step is elongation of transcription. So we know RNA polymerase will move in the 3' to 5' the direction of the coding strand, and as it moves in that direction, it synthesizes the RNA in the new RNA in the 5' to 3' direction. So it's synthesized from the 5' end to the 3' end, and as I've mentioned, the bases are determined by the Watson and Crick base pair rules, okay, by the sense strand. Okay, the DNA helix, when the RNA polymerase passes, it rewinds or it closes up after the RNA polymerase has transcribed that chain. And we have seen there's only a short duplex, after which the RNA detaches from the point of synthesis. So elongation continues until we reach the termination sites. So again, similar to how we saw at initiation, there are specific DNA sequences which function as a signal of termination. Okay, and this termination, we can classify them as being row dependent. This is a protein called row dependent or row independent. Let's first look at row independent. In the row independent terminators, they have several features. One of them is that the sequences on the DNA that form the RNA, self-complementary sequences, allow a hair loop structure in the RNA. So what does that mean? It means the sequences, the codons or the bases on the template strand, when they are copied, okay, when they are copied, when the RNA detaches from them, it is able to base pair within itself to create a hair loop structure. Okay, and then as we continue in this in that template strand, we find conserved AA residues which will be transcribed into UUU in the RNA. And we know the base pairing between adenine and uracil is not very strong. And so that means this strand will now easily detach from the DNA. Okay, it will easily detach from the template strand and it will be released, and then the RNA polymerase will release. So the row independent terminators will do two things. One, will have sequences that allow formation of a hair loop structure in the RNA that has been formed. Okay, and then at the end of this hair loop structure, we'll have conserved 3 adenine residues, which will code for a UUU. And as I've mentioned, the AU binding is not very strong, and so it will be easily detached, allowing the double helix to close back again, and the detachment of the RNA polymerase, signaling the end of transcription, the termination of transcription. So this is row independent termination. The other termination is the row dependent termination. So who is row? So row is a protein which is an enzyme with ATPase activity and also with helicase activity. Okay, so here we can see elongation we have continuing. We are having the RNA polymerase bound to the double helix, and we can see here the replication bubble, and we can see the newly synthesized RNA being formed. Now, sequences or genes that depend on row-dependent termination have a site called the RUT site, or row independent site. Now here, this codes for a sequence that will be there on the RNA that can allow the raw factor, the raw protein, to bind to the RNA. So you can see this is RNA has just been transcribed, and it has transcribed these sequences which is found at the RUT site. And so the raw factor can bind to the RUT site. So on the RNA, remember, not on the DNA, which has been transcribed. Now using its ATPase activity, the raw protein is able to move down the messenger, the new RNA that has been synthesized, until it reaches the RNA polymerase, which has posed at the termination site. So using its helicase activity, this protein is able to separate the temporary RNA-DNA hybrid. And what does this mean? It means now the RNA can detach from the DNA, and when it detaches, the RNA polymerase detaches, and the process of transcription is terminated. So one of the differences is that termination in the row independent does not require the raw factor or the raw protein, but the termination here in the row dependent termination requires this raw protein to use its helicase activity to detach the RNA hybrid detach the DNA hybrid helix. And so this will eventually cause detachment of the RNA polymerase and the RNA from the DNA, so it can go back to its double helical structure. So if we look at the regulation of transcription, because transcription is an important process that provides us with RNAs for different purposes. So it can be regulated to form the transcription, the RNA the transcription is highly regulated to form gene products that are only needed by the cell in proportions needed by the cell. So there are various mechanisms that are involved in the regulation of transcription. One of them is that we saw variations in promoter sequences. Remember we mentioned we have the UP sequence, which is only present in highly transcribed genes. So if a protein is being required a lot, it's used continuously in the cell, you'll have an upstream gene which will increase the rate of transcription. Others, the protein binding really depends. We have protein binding which can either activate or repress transcription of a gene. If we look at this example, we are looking, this is from E. coli. We have a gene that codes for lactose. Lactase enzymes, one of the lactase group of enzymes that is involved in breaking down of lactose. So if you have a bacteria that is growing in an environment where there's both lactose and glucose, we're going to see the RNA polymerase enzyme will be able to bind to the promoter and express the co-expression of the protein or the enzyme that is involved in breaking down lactose. Okay. Now, if we are going in another environment where there's lactose, but there's no glucose, we have an activator protein called such as CAMP receptor protein, which you refer to as CRP, which increases transcription of enzymes that metabolize metabolize glucose, that metabolize the breakdown of sugars other than glucose. Sorry, here there's an error. This metabolize the sugars other than glucose. So here is our good example. Here we have lactose, but no glucose. So that means we'll require the enzymes for breakdown of lactose. So what does mean? Lactose will bind to this site and activate transcription of these genes, okay? Such that we can see there's more binding. It facilitates the binding of the RNA polymerase on the genes for synthesizing of this enzyme. So we can think about the CRP receptor protein as an activator protein. But what happens when the bacteria grows in an environment where there's no lactose, but there's glucose? Okay, so we have repressor proteins such as LAC repressor, and LAC repressor will bind to its site and inhibit the transcription of this gene that is necessary for breakdown of lactose. Okay, so we can see here good examples of how transcription is regulated dependent on what is available. Remember we said proportions needed by the cell. So if there is no lactose in the environment, we do not require this enzyme that breaks down lactose. And so this transcription of this gene is inhibited by a repressor protein, for example, the LAC repressor. Where we have an environment where we have that particular sugar, lactose, we require increased transcription of this particular gene for enzymes for breakdown, okay? And so we'll see that the CRP will increase expression. But when we have glucose, we prefer more expression of both lactose and glucose. So we have expression occurs, but not at a very high concentration because we prefer glucose as our source of sugar. So this is how regulation of transcription occurs. So finally, as we come to the end of the class, we want to look at differences in transcription between prokaryotes and eukaryotes. We know that the prokaryotes do not have organelles, and so we find that transcription and translation both occur continuously in the cytoplasm. However, in eukaryotes, the DNA is located in the nucleus, and so transcription will occur in the nucleus, and then translation will occur in the cytoplasm. So the detranscribed RNA has to be carried out into the cytoplasm for translation and other processes to occur. Then another difference occurs in the initiation of transcription. We saw in prokaryotes this was very simple as DNA is not associated with proteins. However, transcription initiation in eukaryotes is slightly complex because we have DNA associated with histones, and these histones also regulate initiation of transcription. Another difference is that in the RNA polymerase, we saw in prokaryotes there was only one RNA polymerase that synthesizes all the types of RNAs that are required. However, in eukaryotes, we have three different types of RNA polymerases. RNA polymerase 1 synthesizes ribosomal RNA. RNA polymerase 2 synthesizes messenger RNA, while RNA polymerase type 3 synthesizes transfer RNA and 5S ribosomal RNA. So this is a major difference between transcription between eukaryotes and prokaryotes. Another difference is binding to the promoter. We see that the RNA polymerase has a sigma subunit in prokaryotes that is able to recognize and bind to the promoter region of DNA. So this binding is direct. However, in eukaryotes, the RNA polymerase does not bind directly. It requires some proteins which we refer to as initiation factors that facilitate the binding of the RNA polymerase to the DNA. And we learned about the promoter regions. One of the major differences is that prokaryotes have the Pueblo box, while eukaryotes have a Tata box. And this is in addition to other promoter regions we are going to see, like we saw the -35 in prokaryotes, while the eukaryotes have the INR sequence. Then post-transcription, after the messenger RNA is made in prokaryotes, it can easily be translated. So we don't have any post-transcriptional modification. However, in eukaryotes, the messenger RNA is not ready for translation. We have several changes that happen, including splicing, poly-A tailing, and RNA capping. Let's look at some of these. So we are saying that eukaryotic cells, they modify their RNA after transcription. So the enzymes in the eukaryotic nucleus are able to modify the pre-messenger RNA through a process called RNA processing before the complete RNA is sent to the cytoplasm for translation. Okay, so some of the changes that happen in this pre-RNA, one of them is that the 5' end of this messenger RNA receives a modified nucleotide, which is referred to as the 5' cap. And the 3' end gets a poly-A tail where many residues of AAA are added, of adenine are added. These are referred to poly-A tail. So what is the importance of these changes? One, they're able to facilitate movement of the messenger RNA from the nucleus to the cytoplasm. And we know that the messenger's RNA or the RNAs don't stay for a long time in the cytoplasm. So they're able to protect the messenger RNA from hydrolytic enzymes. And they also help the attachment of the messenger RNA to ribosomes, especially the 5' cap. Okay. Another change we say that happens post-transcriptionally in eukaryotes is RNA splicing. So what interesting thing we find about eukaryotic genes is that the RNA transcripts have both coding and non-coding stretches of nucleotides. Okay, so the coding region are called exons, expressing regions, while the introns are the intervening regions, do not express. Okay, so these non-coding regions need to be removed, and they are removed by the process of RNA splicing. So for example, here you can see our pre-messenger RNA is very long, but after we splice and remove the introns, it becomes shorter. So this is the mature messenger RNA, which is ready to be transcribed and to be translated at the RNA. Okay, you can see the 5' cap here and the poly-U tail. So we know the process of RNA splicing removes these intervening sequences or the introns. So how does this happen? So this process of RNA splicing is done by a group of RNAs called spliceosomes. So the spliceosome consists of a variety of protein and small nuclear ribonucleoproteins or snRNPs. So the spliceosome has the interesting characteristic that is able to recognize which has the splice sites. Remember, these are just A, G, T, C, but the spliceosome is able to say, oh, hey, this is an exon, this is an intron. So it is able to recognize them and bring them together and splice them out and synthesize our mature messenger RNA. Okay, so you can see here is a good example. We have our gene on DNA, which contains both exons and introns. This is transcribed into a pre-messenger RNA. Then we have our 5' capping occurring. Okay, then we have our splicing, and the 3 exons are brought together. This is what is done by the spliceosomes. And then finally, we have the polyadenylation at the 3' end. Then we have our mature messenger RNA, which is now carried to the ribosomes for translation. So the process of transcription has several clinical correlations, as several antibiotics have been found to inhibit the process of transcription. For example, rifamycin or rifampicin and streptobaricin, they're able to bind to the beta subunit of the bacteria polymerase to block the initiation of transcription. We now use here rifampicin for the management of tuberculosis. Another antibiotic, antinomycin D, is able to form a complex with a double-stranded DNA, and so this prevents the movement of the RNA polymerase, and so it inhibits the process of elongation. And then we have streptolidine, which is again able to bind to the beta subunit of prokaryotic polymerase, and hence it inhibits the process of elongation. So the difference between rifampicin and streptolidine, this one inhibits initiation and this one inhibits elongation, although they bind to both beta subunits. So this is the end of our lecture on transcription, and let's meet as we talk about translation. Thank you.